Hello guys, today we're going to learn about DNA, its structure, its function, and the importance of all this. So, right here we have our basic DNA strand. And of course it's not laid out in a double helix formation like we're accustomed to seeing. It's more flattened out so we can get a conceptual idea of what's going on. So, right here we have a deoxyribose, a sugar, then a phosphate group, and it staggers depending on if you're going up or down. And same for the other side, phosphate deoxyribose. This is considered your backbone of your DNA molecule. Inside, we have a cytosine, C, guanine, G, adenine, A, or thymine, T. These are your nucleotides. So together, they just come together and make your DNA strand. If you have a deoxyribose, label it 3'. And if you have a phosphate, label it 5'. So we have a deoxyribose, a phosphate, 3', 5'. Your backbones will always match up in this manner. It will never be a 5' to 5'. Or 3' to 3'. It's always 3 to 5. And same thing for the top. You have a 5' up here, so automatically think 3' up here. All these are connected through a phosphodiester bond. So all these bonds, phosphodiester bond, keeps it all glued together. Now we're going to jump in to replication. And important words will be posted on the notes section of this video. So you don't have to worry about spelling, and you'll all have your notes. So the step 1 of DNA replication is a helocase comes in. So here's our very, very crude DNA molecule or DNA strand. So this helocase comes in let's put him right here in this location. So it comes in, it attaches and begins to make a bubble. So right here at our bubble, we're going to draw a line. And it splits the two. It doesn't split the two DNA molecules, but it puts them in two halves where replication can begin on each segment. So your helocase just makes a little bubble in your DNA so it can begin the replication process. Now, after the helocase comes in, we have to figure out which side is the leading strand and the lagging strand. And this is where the importance of the 5' to 3' come in. So, say we have 5' up here, 3' up here, or down here. And so since this end is 5' and we go all the way down, this side would be a 3'. Same thing right here. If this is a 3', this will be a 5'. So, since our helocase would be right here making the bubble, and our DNA strands go from 3' to 5', we need to add a DNA RNA primase to this point. So, since this is 3', this would be our 5'. So, our RNA polymerase is right here, and it starts the adding, it initiates the adding of nucleotides. The nucleotides aren't being added yet, but it hollers out to DNA polymerase to come attach to it. So, here's your DNA polymerase attached to your RNA primase, and the DNA polymerase runs all the way down. So he will go all the way down this strand and add nucleotides. So we have a new DNA strand. This end right here is called your leading strand because it just keeps on going. You don't have to worry about any kind of stopping point because it's going from 5' to 3'. So now that we have that, we're going to get rid of this real quick so it's not distracting. And say we are up here. Now I'm going to extend this a little bit more. If we start right here, it wouldn't be 5' to 3'. This would have to be a 5. This would have to be a 3'. So your RNA primase cannot connect to this point. So it would have to come connect to here. But then it would add nucleotides this way. But what about this? So what happens is RNA primase comes and then it attaches at various points within your DNA strand, your lagging DNA strand. And that's what's called lagging, is because it has to go to various different points. So there's your RNA primase. Here is your DNA polymerase. And each one of these runs down, adding those nucleotides as it keeps on going. All right, so each of these segments are called Okazaki fragments. And we can see why. It's because they are fragmented all throughout the lagging strand. Now we're going to act like we had two new DNA molecules. So this will be our, our leading strand. I cannot draw and think at the same time. So this is our leading strand. Now this is our lagging strand. So since this is our leading strand and that's our lagging strand, there's something that wasn't really in the drawing from last time. It would be our RNA primases. So we need to get rid of those. They are not needed in the function of DNA. So exonuclease comes in and essentially just removes the RNA polymerase and replaces it with a nucleotide of whatever needs to go there. So if an A was here, then a T would go there. So it just replaces the corresponding nucleotide. You'd think we'd be done, and we're not. So now that we have those strands, it has to be proofread for mistakes. If it's not proofread, then a mutation can happen. And we'll learn about mutations later on. So what happens next is DNA ligase comes in. And it's really hard to draw. So this is where imaginations come in handy. DNA ligase comes in and creates the phosphodiester bonds. So the DNA ligase comes in and creates the phosphodiester bonds. And now we have two new DNA strands copied off of the original strands. So what's the importance of all this? This is where we get our genetics. This is how we became to be here. All our cells replicate. Every cell in your body will replicate except your neurons replicate when you're younger, but then they stop. So your cheek cells constantly replicate. Your stomach cells replicate at an astounding number because they're subjected to acid. And even your gametes, your sex cells, your sperm and egg, they have to reproduce. So that's where DNA replication comes in. The DNA sends signals to your cells saying, hey, we need to replicate, or this needs to be made, this needs to be done, and that's it. So tomorrow, what you guys need to bring is spit. We are going to do a little worksheet over all this, and then we're going to slush some salt water and gather up some cheek cells and capture our own DNA, and it'll be in little tubes you all can take home, so it's going to be fun. So have a great night and